I really would like to express my deep gratitude to all my dear teachers who have taught me so much, and all my dear friends with whom I feel the world is so warm, and this evening it is really wonderful. And all my partners. Nothing would have been done without your help. I would like to entitle my short presentation A Late Comer and Still and Forever a Learner. BGI was really born for the participation in the Human Genome Project. We began with nothing, or almost nothing. As one of our friends described, the building had nothing else except for a nice double helix. I really wondered if we could get the support to become an internationally competitive group. It was true. It was almost unimaginable to have such a mission to sequence 500,000 reads, or about 1% of the human genome. One of our US colleagues has written in this way: Human Genome Project. That work was already well underway in other countries like USA, UK, and China was way behind the curve. But that guy from China was really earnest and convinced him that China's involvement would represent a major advance for China. And then BGI, formerly Beijing Genomics or Genome Center, came on board. We did the job, even compared to the great contribution by USA, UK, Japan, France, and Germany. Our contribution could not have been even smaller, only 1%. Of course, actually, we did a little more. We contributed the 64 megabytes of the raw data to this paper in Nature. 64 megabytes, approximately 2% of the human genome. Our job. That's the first time we have the opportunity to prove ourselves was acknowledged by the international community as well as the Chinese authority, especially our colleagues. I would like to quote from James D. Watson. This center in Beijing, led by a Chinese, now compares and competes well with equivalent centers elsewhere. This message has never been misinterpreted by us. It was expectation. It was a hope on us. That's really true. We gradually grew at that stage of single sequencing technology. We moved from Beijing to Hangzhou, a city on the southeast of Shanghai. Perhaps this paper has been forgotten, but this is the first time a publication by Chinese mainlander as a cover story by Science, the full cover. Science says something like that. Please allow me to quote again. This team deserves enormous credits for their outstanding world-class accomplishment in a remarkably short time. It is true. It took us just 74 days, and then we have done the job just exactly according to our plan. Science magazine, the major chief editors came to Beijing to host this press conference. Science magazine ordering China's sequencing of the rice genome. This paper has been cited many times, about 2,000 citations now, And our database, with the free access of the rice genome data, has been regarded as one of the most popular databases in genomics. And we can see from this figure, before our publication and the release of the rice genome sequence, there were much more researches on the wheat than on rice. But after that, there have been more on rice than on wheat. If we put it more professionally, we can see the difference between the number of cloned genes and the QTL of rice and of wheat. That significant difference was really made by release and the publication of the rice genome. From this example, we can see the potential of the human genome sequence. It will make even bigger or greater contribution to our health. We have to be patient. And after that, if we say the draft sequence was not well cooked, and now we have the right genome well cooked, this is a cover designed by ourselves. I like it. You know, nobody can eat more than a bowl of rice, but we love to make so much to share with all those who like it. That's still encouragement from John Watson. But sequence is a significant part of the human genome, as well as the entire genome of rice. China is now a leading genomics nation. This message has not been misinterpreted either. That's an expectation. That's encouragement. Even now, I could not agree with the last sentence of that, how to say, movie of advertisement style. We are not leading the way yet, and then we would not intend to lead the way. And now came the third stage, if I might call it. We moved again from Beijing to Shenzhen, the twin city of Hong Kong. We were growing. We have been growing from 1% to the human genome, and then 10% to the HappyMap project, and then we completed the first Asian genome. Please don't ask me whose genome it is, because now we are still discussing the ownership of one's genome. Technically, it's important. It demonstrates the methods available now. The now generation or new generation sequencing technology are extremely powerful. Science should not wait. We should not wait. We have to take the opportunity. We have to work even harder. That's the reason we have proposed that the concept of a human pangenome, which was published in Nature Biotechnology, that our genome is composed of two parts, a common part, as indicated by the Human Genome Project, and some extra part which cannot be found in the reference genome sequence generated by the HGP. We worked in order to reveal the possible interaction between genes and the environment. That's just one of the examples which was published in Science, that one of our genes, which has a quite different frequency in high Chinese who live at the altitude as high, as low as we live, and the Tibetan Chinese who have been living in the wood roof. And then we also have demonstrated that now sequencing technology to sequence the exomes or the whole genome sequence encoding protein are very powerful, and then we really can find more. We have more than one genome. There are a lot of genomes in our body we call metagenomes. We have published the first catalog of human gut metagenomic map to indicate there are numerous, at least 2,000 species of genomes, and there would be 150 times that of the human gene. Those are bacterial genes who are helping us, who have been together with us. We also have sequenced the trace DNA or partially degraded fragments. For example, 4,000 years ago, we recovered more than 80% of the genome. It means we were able to sequence trace DNA or RNA. Panda is so cute. We sequence it, of course, together with our colleagues. Technically, that's the first successful de novo assembly of a man genome without the help from a genetic map or physical map. We published the first draft genome sequence of silkworm because it is useful, but now, with the new technology, we have sequenced much more, at least 50 strains, both wild and domesticated. We have to know more. For example, our social behavior and neurobiology. We sequenced 2 species of ants. They are so different in their social organization. We found, at least, that the queen was not born as a queen. And then we also worked on the new version of the maize genetic map, maize as a classic model for genetics. Now, by sequencing, we revealed much more about it. Darwin loved cucumber. Why don't we sequence it? We did it, and then also soybean, because most, if not all of the Chinese do love bean curd. We are still at the beginning. We are not really ready, but now we are going globally. Please don't ask me where I am heading for. Don't you see clearly that the road is exactly beneath our feet?